Good afternoon. I'm Beverly Edmond, and I have the privilege of serving as the interim provost and vice president for academic affairs here at the University of Montana. I am so delighted to welcome you to the first of this year's Provost Distinguished Faculty Lecture. This lecture series celebrates the University of Montana faculty who have been nationally and internationally recognized for their scholarly achievements. Its purpose is to share the exciting accomplishments of our faculty with the broader Missoula campus and communities. This presentation is being recorded by Missoula Community Access Television as a part of a media assistance grant donated by MCAT. At this point, I'll ask Dean Comer if you'll come forward and introduce our lecturer this evening. Thank you.
Thanks, Beverly. It's always a pleasure to be here for this particular reason. We have some amazing faculty, and it's really so important to stop once in a while and just take notice of what they've been up to. As a scientist, I don't know about you, but I always like reading papers that have opening lines like, it's really important that we understand how the brain creates memories, or it's really important how we understand the complexity of cancer, or it's really important how we understand how societies create communication webs, something really fundamental. And then the research systematically goes after that. And our speaker today is somebody who has taken on a really big question, the kind that we like to talk about here on campus, which is concern for the environment, concern for how global climate change is impacting the Earth. And the logic is really impeccable because with the Earth about 80% covered with water, it's an incredibly important surface for chemical reactions that are going to affect the whole planet. And so Mike de Grand Prix and his colleagues have taken on the issue of trying to come up with a quantification of what's going on there. And as everybody who's a scientist knows, if you want to control something, you have to be able to measure it first. And so that's where an analytical chemist really comes to the fore. So Mike got his PhD from the University of Washington in analytical chemistry. He then spent a period of time, which I bet was wonderful, at Woods Hole, which is on the edge of Cape Cod. Great place to be. And then he came here in '96 and joined the Department of Chemistry and Biochemistry. And Mike's been a stalwart citizen of the university and the college. He's been on lots of committees and done lots of really wonderful things for us. So you need to know that he'll cover the science, but he's really kind of a multi-talented guy and has a lot of dimensions. One of those I particularly like is if you check him out on the web, you'll find out that from his grants, he's produced some really wonderful outreach for kids, projects that demonstrate how climate change is actually affecting the oceans, so that kids can see it firsthand right in front of them. Something that we didn't do 20 years ago in science, but now we do, and Mike's done a really great job with that. He'll tell you about these wonderful devices he's built and what they all mean, but it's really a pleasure to introduce Mike de Grand Prix, a good friend and somebody who really represents us well. Mike?
All right, thanks, Chris. I guess you can see where the sewer pipe in the title came from. This is the first instrument that we developed. So today I wanted to take this opportunity to talk to you about the development of our CO2 and pH sensors. One of the things that I wanted to focus on is that the process over which these sensors were developed was a really long time frame. And so hopefully through this process you'll get an idea about the process of invention, a better understanding of optical chemical sensor technology, and also the role that entrepreneurship can play in a process like this. I'll talk about the background of this slide later. Okay. So today I'm going to give the motivation behind the development of these CO2 and pH sensors for aquatic research applications primarily. So this process started in 1990 when I became a postdoc at Woods Hole Oceanographic Institution. And so it's been over a 25-year process of first inventing the device, and it started out with this sewer pipe right here, and then having that evolve, even to the present day, it's still evolving. So I'll talk about that whole evolutionary process. And then it culminated in 2015 with the award of this XPRIZE competition, which if you haven't heard about, I'll fill you in on what that's all about. And then I'll touch a little bit about current directions, what we're doing right now. So, I'm going to get the handle of this here, back. So the upper left plot there, most of you are familiar with. It's the atmospheric CO2 record at Mauna Loa Observatory since 1958. And you can see that increase in atmospheric CO2 due to anthropogenic CO2 from fossil fuel combustion and biomass burning. So that CO2 is over 500 parts per million now, or 400 parts per million now, and it's continuing to increase, but that level would be much higher if the oceans and the terrestrial biospheres hadn't absorbed a lot of that CO2. So you can see here that about 55% of the anthropogenic CO2, which comes from fossil fuel combustion and biomass burning, is accumulating in the atmosphere, but a good portion of that is actually going into the oceans, about 30% of that. So if the oceans weren't taking up that anthropogenic CO2, the planet would be about 1 degree centigrade warmer than it is today, based on just its effect as a greenhouse gas. So it's having an important ameliorating effect on the accumulation of CO2 in the atmosphere and greenhouse warming. However, there's always some kind of consequence in a situation like this. And so the oceans, their oceans chemistry is changing because of this absorption of CO2. Our oceanographers had the foresight to start these long-term time series back in 1988 at a bunch of different locations globally, or a handful of locations. But Hawaii and Bermuda are two of the prime locations for these long-term measurements. And so they go out in a ship every couple of weeks and do a whole suite of different chemical and biological measurements. And what they found is that this is the CO2 in parts, well, microatmospheres, which is equivalent to parts per million. From 1988 to 2010, the red dots are the mean levels, and these bars here represent the seasonal cycle, the range annual cycle at each year. So this is the increase in CO2 due to the increase in atmospheric CO2. So that's the CO2 going into the surface ocean. Same thing at Bermuda, at this Bermuda Atlantic time series over here. Going with that, though, is a drop in pH. So what happens when CO2 dissolves in seawater or in any water, it forms carbonic acid. That's this reaction right here. CO2 reacts with water, forming H2CO3, which then, as an acid, it will drop the pH of water. So as you increase acidity, pH goes down, and that's what's been documented at these two sites. This process is called ocean acidification. And it's important because the mean pH levels have been constant for hundreds of thousands of years in the oceans. So humans are actually affecting mean pH levels in the surface ocean. So this has important consequences for organisms. This guy is not very happy, but they've done a lot of different laboratory incubations at high CO2 levels, different pHs, and they've shown things like fish are affected, shellfish can be affected, a whole range of different organisms can be affected by this dropping in pH. Particularly shellfish, because their shells, their solubility of their shells is dependent upon a static or higher pH level, and so those shellfish organisms are the most affected. For this reason, we need to understand the carbon cycle really well. We need to understand why or how the oceans take up atmospheric CO2 and what controls that process. And we need to know the consequences of that, what that CO2 is doing in the surface ocean, how that's affecting ocean acidification. So the way oceanographers have traditionally done this, they've gone out on a ship, they've lowered a water sampler into the water column. This is called a hydrocast. They drop this water sampler deep down into the ocean, then bring it back up, trigger the bottles at different depths, and then they take samples They gather, it's a scientific ritual. They put on their rubber boots, they gather around the CTD rosette is what it's called, and they tediously collect these samples. Then they take them back into the ship's lab. They either archive them and take them back to land for analysis, or they measure them right on the ship. So this is studying the oceans one sample at a time. And it's very time-consuming. It's very expensive. Having 30 scientists on board a ship for a month at a time is an expensive endeavor. So there's alternatives, things like tricorders. So ideally, we would have something we could wave around and say, wow, this planet's CO2 flux is 3 gigatons of carbon per year, and they're in trouble. Or have something like that. Well, satellites can measure properties over wide spatial ranges, but nothing in this, no chemistry in the surface ocean at present can be measured via satellites. So the other alternative is to actually use sensors on a buoy. So we can put autonomous sensors out and leave them and collect continuous data to help us understand the carbon cycle more effectively. So instead of having a ship that goes out there and just gets a snapshot of information, we can get continuous information. When I first started proposing this idea about CO2 sensors, there were no CO2 sensors when I came into this in the early 90s. My reviews would say this often, something like this. This is the holy grail of ocean sensors. So there was a tremendous amount of interest in this at that time, and there was nothing available. But it's not easy to develop sensors for oceanographic applications. They need to be accurate, and that means that you calibrate the instrument on land, then you deploy it, and it cannot drift. It cannot lose that calibration. It needs to remain accurate. That's because it's very difficult to calibrate an instrument when it's already deployed out in the ocean. That adds a lot of complexity. You have to have standards. You have to have known concentrations you can calibrate the instrument with. It's not practical to do that. So you need a measurement that doesn't drift. You also need sensitive and precise measurements. So most concentrations in the ocean are very low, and they also have very small changes on that background. And so you need to be able to detect low levels and also detect small changes in those levels. So they have to be sensitive and precise. And then reliable. They cannot be too complicated because they're more likely to break. They might be more power hungry and more expensive as well. So there's nobody out there to fix them. And if they break, you don't get any data. So the reliability is crucial. And low power. Power is important. You cannot put solar panels on a buoy. Typically, it's not that feasible or not readily doable. You cannot put big battery packs in the ocean. They're too cumbersome, too expensive. So you want things to be low power. If you can meet those first 4 criteria, then the low-cost one is less important because you're willing to pay for these nice features. But obviously, low cost is important because then you can buy more and spread them out throughout the world's oceans. So ideally, you would have a sensor that, oops, let's go back. A sensor that looked like that, a simple sensor, the probe that you could stick in the water and stick on a buoy and leave, and it'd be low power and collect really accurate data. This is just a pH electrode, which actually do not work well in the oceans. But back when I was first getting involved in this in early 1990, the only sensors like this that worked in the ocean were dissolved oxygen sensors, and they were pretty widely used. They were good sensors, and they've improved a lot since then, but those were really the only chemical sensors that were used in the oceans in 1990. But one reason why is because people cannot make sensors like that with all of these features. And so the danger is creating a one-eyed monster, I call it, a one-eyed monster, sensor monster that is too complicated. It's hard to understand how to use it. It's not reliable. It's too expensive. And so throughout this whole evolution of these sensors from here to here, we've been trying to avoid becoming the one-eyed monster. And so you can be the judge of whether we succeeded in that or not. But back before I came along, there were already CO2 sensors, optical CO2 sensors that were reported in the literature. In the 1980s, fiber optics were first coming out. And so everyone was interested in using fiber optics and putting chemicals down on the end of the fiber optics and measuring different materials, substances, different chemical species. CO2 was readily doable that way because you could, the sun just came out and blurred my screen, but so here's the sample with the CO2 in it. It can diffuse through a gas permeable membrane because remember CO2 is a dissolved gas like oxygen. And so it can go through this membrane. It can then, this is your sensor side of the membrane. It forms carbonic acid and hydrogen ions which decrease the pH. And so if you have a pH indicator in that solution with it, you can look at the color changes and determine what that CO2 level is. And so the way that might be done is, this is a pH indicator spectrum, absorbance, and wavelength. And I'll explain this because I know a lot of you have never seen data like these before. So the higher the absorbance, the more light the indicator absorbs. This pH indicator is metacreosol purple, that's the name of it. But it has an absorbance, the one form has an absorbance at about 580 nanometers, and another at about 430 nanometers. This is, this light at 580 is yellow-orangish, and this is violet-bluish light. So those are the two colors this indicator can absorb. At this form is in the basic form. So when there's low CO2 in here, this is low, and it's more basic. This will be more abundant. As more CO2 comes into this solution, this form will shift to this more acidic form. So you can look at these wavelengths and determine what the acidity is of the solution by monitoring those wavelengths and looking at the intensity of light that's transmitted to these solutions. So that's how these sensors existed back in the 80s, and they look like something like this, where they would put a membrane down on the end of fiber optics and shine the light down into this reagent behind a gas permeable membrane. These were called fixed reagent sensors. They drifted significantly because that reagent chemistry wasn't stable over time. Different contaminants could get in there, or the indicator could become photo-bleached because of the light intensity hitting it. So there are lots of drift mechanisms for these sensors, and they never were very stable, and then they still haven't proven to be stable. So here's an example of that sensor with the light actually on, and then the light comes back up to your detection system. So that's sort of the ideal probe-like geometry that we all want to strive for. But when I was a grad student, some other students were at University of Washington, some other students were working on a sensor that actually renewed the reagent. And so what this does is this is about a 1 millimeter diameter membrane here. We put in tubing and it pumps down the indicator down into this hollow cavity, and then it moves out this exit right here. The light is shined down into this membrane, and some of it's scattered back up another fiber optic back to your detection system. This had never been used for CO2 before, and I proposed to do this when I went to Woods Hole Oceanographic Institution as a postdoc. And they liked that idea, so I got the position, and we actually built that. This was the first sensor I built in the early 90s. That's a quarter. The white membrane is in here in this little cap that I have over it. This actually worked reasonably well. And I took this out on a cruise. I was lucky enough to go out on a research cruise from Hawaii to Tahiti for 18 days. And I took this sensor with me. It was just on the ship, though. We didn't put it on a buoy. This was just a preliminary test of the system. And I got this first paper out in '93 demonstrating the potential of this technology. This was a bubble trap, though. And so oftentimes air could get in here, and then a bubble would never leave. It would get stuck inside of in front of these fibers. Well, bubbles and light don't mix. The light transmission will be scattered, and it will be very noisy, and you don't get good measurements. Alternatively, we did this flow-through design where we came in with one tube. This is a small capillary tube. Pumped the indicator into here. Now again, CO2 is freely diffusing across here, equilibrating with that indicator solution, changing the color of it. And then that solution is pumped up into this optical cell. This was much better in terms of bubbles. There was one insurmountable problem between these two sensors, though, and that is that they were both required very high-intensity light sources. Very little of the light coming out got back into this return fiber optic. And so we needed a very high-intensity light source for these types of sensors. Consequently, they weren't practical for an autonomous instrument. You needed a projector light intensity bulb like that up there, 100 watts, and it's just not feasible to have an instrument like that. So I gave up on this design, and it took me a long time to accept that, but working with another engineer, we developed, oh, here's a picture of actually one of these sensors. And this is a silicone rubber membrane that's gas permeable, so the CO2 could diffuse across that, and then the fiber optics are back in here. We went to a transmission geometry. Instead of the fibers coming down like this, we went to a conventional design where the fibers were facing each other. So instead of having a 100-watt bulb, now we had a little white light source, which is like a pen lamp sized light source. And a couple of orders of magnitude lower in power requirements. And so the light then is the indicator, equilibrated indicator solution in the membrane right here, comes into this optical cell, and the light's transmitted across there. We look at the color of it at these wavelengths, and then it exits out the flow tube there. This worked very well, and this was the basis for the CO2 instrument that we ended up developing, and we still use a design very much like this, and I'll describe that. This is what it looks like on the end cap of the instrument. So the fiber optics dictate a lot of this geometry. This is about 6 inches in diameter, about the diameter of the sewer pipe instrument there. And so the fiber optics are here. This is flipped relative to this, but you can see that gas permeable membrane and the optical cell. So other things to describe here is the plumbing design. We have a valve and a pump in this instrument. It's in this little housing right here. That's also on the old instrument right there. That's shown right here. There's a little pump here. We still use these pumps. They pump about 50 microliters per stroke, and we only use 100 microliters for a CO2 measurement. So that's just a couple of tiny droplets of water for each CO2 measurement, because the volume of this whole system is so small. So we don't have to use very much reagent. What this shows, though, is a bag that contains the reagent, which is, for CO2, we use bromethymol blue. That's the pH indicator. And then we also have another bag that has pure water in it, so we can run baseline measurements. A critical aspect of the system is to be able to flush the indicator out and have a solution with no indicator in there and get a baseline measurement. That gives you incredible stability because you can determine that baseline. Without that, we'd have drift that would degenerate the quality of our measurements. So to introduce this periodically, we have a valve in here, a 3-way valve. So you can switch between this bag and this bag periodically. The other thing about working in the oceans is this housing right here. That's this, so the sensor goes right on here. This is an oil-filled housing, because under hydrostatic pressure, the pumps cannot pump against that deep hydrostatic pressure. So the sensors might typically be deployed, the buoy might be up there at the ceiling, and they might be down just 2 or 3 meters. But that's enough that the water cannot push against that hydrostatic pressure. So you have to have an oil-filled housing with a diaphragm on it with oil, so that this has oil inside of it, silicone oil. So this makes contact with the hydrostatic pressure in the water column, and that pump, therefore, does not have to push against that deep water column. So with this would work at any depth then, because this would compensate for the pressure. But without that, you can only work in the very shallow ocean, within a meter or so. Then the optical detection system is another thing we had to develop. This is called a spectrograph, which is in the insides look like this. The fiber optic would come here from the optical cell. It hits what's called a diffraction grating. That they're on your credit cards, that little rainbow you see if they're still on credit cards. Those are diffraction gratings. They spread the light out into their separate wavelengths. And so doing that, we can look at the different wavelengths that correspond to the wavelengths of the indicator that we use at 680 nanometers, 580 nanometers, and 430 nanometers. So we just put detectors right in this plane that detect those wavelengths. And then also we put a reference detector out beyond 700 nanometers where the indicator doesn't absorb light, because we use that for a reference to evaluate the fluctuations in the source intensity. We use a white light source, that little pen lamp in this case. So this is it's about this big, and I'll talk a little bit more about that. So with all of these parts, then we have the optical cell, we have the pump housing, we have the plumbing, we have the reagent bags, we have the detection system. We had all this sitting on a bench. And I was working with engineers at the Oceanographic Institution, specifically one engineer, Terry Hammer, and I said, well, how can we get this in the ocean? He had a lot of experience doing that. He packaged this up in a matter of a month or so, all these different components in a geometry that we could put underwater. That was this instrument. We didn't have a really big budget back then, and so he scrounged up whatever he could find, and this sewer pipe happened to be near his shop there at the Oceanographic Institution, so that's what he used. And so this became we had one of these for 1 or 2 years, and then we built a whole bunch more, but we deployed that off of the pier at the Oceanographic Institution right in Woods Hole Harbor there, and that's the basis for it. Well, you can see it coming back with some biofouling on it there. Biofouling is an issue. And we had the first paper to have in situ measurements of seawater carbon dioxide. We call this PCO2 because it's the partial pressure, but it's just the dissolved CO2 in seawater. So we had the first instrument that measured CO2 underwater. And that was just off the Woods Hole Pier, though. And then, because of these instruments were working well and very promising, we started to deploy them more widely. We put sensors off of Cape Hatteras, and we combined them with oxygen sensors often, because oxygen goes hand in hand with CO2 because they're both part of the photosynthetic process. So they give complementary information. These are all in the 90s. We deployed sensors off California coastal waters. We had a grant to do that. These are data collected in the 90s but published in 2002. This was quantifying air CO2 fluxes on the Middle Atlantic Bight. That's the section of ocean, coastal ocean, from Cape Hatteras up to Cape Cod. And nobody had known the amount of CO2 that was exchanging between those two, between the atmosphere and the ocean in that region. So we were the first to document those air-sea fluxes. And then in '99, we had a paper that demonstrated that these instruments could be calibration free, that the theory showed that every one of these instruments can behave the same, and we actually showed that every instrument could give the exact same response and did not need calibration. This was the basis for a patent that went through the university's licensing system and became, it's still a patent that that the university owns that I'm licensed to use with my company, which I'll describe. But through these papers, we got a lot of interest. I was getting phone calls, would you make us a SAMI, the Submersible Autonomous Moored Instruments? So the Submersible Autonomous Moored Instruments, I forgot to mention that. That's the name of all of these instruments, so the SAMI. So would you make us a SAMI, would you make us a SAMI? And I never thought about being an entrepreneur, but when getting these phone calls, I had to try to do something. I couldn't make these in my lab. My grad students had their own projects, and they were going to become manufacturers. So I made up an ad for the Missoulian, and in 1999, I submitted this ad to the Missoulian, and it read, at least at the beginning, a small business or entrepreneurial-minded individual to build and market chemical instrumentation developed by a University of Montana researcher. And I gave this to the person at the Missoulian, and they said, this isn't for a job, is it? And I said, no. And that person said, well, then you have to put it in the section of the newspaper where work at home and make $500 a week. And so that's where I put it. And I was not very optimistic I would find somebody, but maybe only in Missoula this could happen. A couple of people contacted me, and one person was ideal, David Irwin. He had a small company here making plastic display cases, and it was called Sunburst Plastics. And so he was willing. We had the drawings. We had all the engineering done. We just needed to get the parts and put them together. So he was willing to do that. And we actually then started Sunburst Sensors in 1999. And then subsequently, we got a big grant a combined grant from a few different agencies. And I was able to bring in Jim Beck, a former high school classmate of mine. He was working in Seattle at that time at a biomedical engineering company. And he wanted to come back to Montana, so I was able to bring him back. And then we really focused on redesigning the instrument, making the software more user-friendly, and improving the performance of the instrument in general. So there's a picture of Jim during the XPRIZE competition. So we focused on redesigning this optical cell initially. So I'm going to go through a whole redesign process here a little bit. Blowing that up, you can see that, okay, the liquids come in here and go across here and out here, the pH indicator solution, and light shines across here. Well, these little areas here tend to trap bubbles. And the amount of human hours wasted in pushing bubbles out of optical systems is, if we could just be paid back for that, we would be quite wealthy. But we wanted to improve that, so there's a Z-cell design that was already commercially available, and you can see how this might be better. Liquid comes in here, and it goes right to that interface where the fiber optic is and can flush that. This is much less prone to bubble retention and much less time-consuming in troubleshooting. But the problem is actually making integrating the membrane equilibrator on that type of optical cell. So Jim and we brainstormed a lot, but Jim did most of this work integrating that membrane equilibrator onto this optical cell. And that's what, I'll actually pass this around so you can get a good look at it, but it's a fairly complicated machine device. But all it is integrating the membrane equilibrator with that optical cell. And that's a picture of it that you can actually see it in real life. The optical detection system which we used, this spectrograph with the diffraction grating, was also not ideal. These were getting more expensive. This was a significant cost of the overall device, this spectrograph. And so we wanted to come up with some new ideas on how to remove that. And also the white light source that's used for this required a significant warm-up time. And LEDs are available which do not require much warm-up time. So through a long, tortuous process, we came up with this what's called a beam splitter design. And so what happens is the light from two LEDs, one's the yellow LED for one wavelength, and the other is a blue LED for the other indicator wavelength. They shine, and this is a partially reflecting mirror. Part of it goes to a reference detector so you can keep track of the fluctuations in light intensity. The other part goes through here, out the fiber optic, out to the optical cell. And so we switch between the two light beams, so very rapidly though, so we can keep them separate. So the detection system says, oh, it's the yellow light on now, it's the blue light on now, and electronically keeps those signals separate. So that, this looks like you could buy this off the shelf, but this is a completely novel, not original design, but novel in the sense of what we're doing with it. That looks like this. We actually put the beam splitter, this is all Sunburst's work here, not mine, but we actually put the beam splitter right on the electronics board. And that's this square right here, so the little reflective mirrors here, the LEDs are here and here. And so this is just simulating what's happening, and it's a mock-up. This cell would be outside of the instrument, but this fiber optic would go through to the outside of the instrument where the optical cell is, and then back to this light detector. And so it really simplified the optics. it wasn't much of a leap to take this CO2 sensor, which is that one that's being passed around with the membrane, remove that membrane, and just pull seawater in directly and inject indicator into it. In the CO2 system, we isolate the seawater or fresh water from the actual internal indicator solution by a membrane. So only CO2 controls that pH. But in this case, we want to mix the indicator directly with the water and get the pH of the sample itself. It was not conceptually or even mechanically hard to do that, but it took a few years to actually work out all of the issues with that. But this was the first paper then published in 2008, one of my grad students there, Matt Seidel, showing that in situ measurements, in situ means again in the system, in the water, underwater, indicator-based measurements of seawater pH. So these are the first indicator-based measurements of seawater pH, and you might really be surprised at that, but no one had figured out how to do this in an automated way prior to this instrument being developed. So that led to jumping ahead. This same instrument we've used for years, and then this XPRIZE came along, which motivated us to consider some design changes. And what this XPRIZE was aimed at was two different competitions. One was for very accurate sensors that will give scientists the ability to measure ocean acidification throughout the world's oceans. And then also an affordability competition for affordable and easy to use sensors that will provide the tools necessary to track and respond to ocean acidification. So we entered both of those categories with two different instruments. The prize for these was $1 million in each category, $750,000 for first prize in each category, and $250,000 for second prize in each category. This is, we call this the titanium SAMI. It says throughout the world's oceans for this accuracy purse. So this had to go deep down into the oceans and function. So we needed a titanium housing to do that. That was a pretty simple modification of our existing SAMI. The affordability SAMI, though, notice there's no cell up here, fiber optics. We used an integrated flow cell design for this. This is much more of a big step forward, and I'll describe more of that later. But this is the inexpensive SAMI. It can only be deployed in the surface ocean, but in principle it's a lot less expensive, although we're not selling these yet. So a whole bunch of teams entered. 16 actually showed up with instruments. There were a whole bunch of different technologies. Colorimetric is the indicator-based ones. We had some competitors with using our own technology. A bunch of different electrochemical methods. Not to go into any detail there, but there were about 16 different companies, and then more than that in terms of instruments, because we showed up with 2, for example. This is back to that original slide. This is the background slide in my first slide. This is a test tank at Monterey Bay Aquarium Research Institute in Moss Landing, California, where the initial XPRIZE testing took place. And this is I believe, the inexpensive SAMI, and this is the titanium SAMI. It looks like a cryogenically preserved sensor facility. I don't know, it looks strange, but it's all they are is they're just dropped down into 5 meters of water there, and they're dangling and taking measurements, all of them at the same time. And so that was the first place where we tested things. Then we went up to the Seattle Aquarium. They built a channel, a flowing seawater channel, where all the instruments were placed for about 3 weeks. And meanwhile, the XPRIZE people were taking samples and doing comparisons with all the different instruments. Then Jim went to Hawaii and took the titanium SAMI, and they deployed that off of a ship down to 3 meters deep, and it worked. We had never deployed the instruments that deep before, so we didn't know if it would work, but the titanium held up, and apparently we got good data because we won both categories of the XPRIZE. So it was a little weird in that the final tests were done in May in Hawaii, and then the actual award ceremony wasn't until the middle of July. And it was sort of like a beauty contest because all the contestants had to show up, and they opened the envelope, and the winner is. And basically, we didn't know if we'd won until we were on the stage. And so this is that stage where we were. It was in New York City at this nice venue, But this is Wendy Schmidt, and she's the philanthropist. She got her money when Google went public, and so she's a multi-billionaire. But she's very interested in ocean conservation, helping prevent the degradation of oceans, and so she sponsored this whole XPRIZE competition, and that's Jim and Jim's wife, Kitten, and two guys from XPRIZE. I'm not going to remember their names, but so this then has led us to further research and development of the SAMIs. So this one, remember, does not have the fiber optics up here. So what we did is we integrated the optics with the flow cell, and I'll show you that. But so we've gotten away from this fiber optic mess on the top of the end cap almost. And so what we've done, and Ruben Darlington, the engineer at Sunburst, is the key guy in doing this, is we've integrated this beam splitter. These are the optics. This is the light with this flow cell. We basically brought those together. And it looks like this. It seems trivial. You look at this and say, okay, here's the beam splitter, here's that Z cell. These guys just put them together. But in inventing something like this, you can never figure out to do this combination if you didn't do that both parts first. So really, it's a sequence of inventions where you have this new invention and you say, well, okay, this is working. Now what can we do next? To actually come up with this design originally without having that stepwise process would be very, very difficult. And so that's a part of that inventive process, is that you build off of your prior inventions. And then this is just a drawing of that of this instrument. So this is these are all pH now. These are not the CO2, but this instrument is where we're about the size of what they should be here. We're not quite done with it, but we're still waiting on electronics for it here. There's the pump and the valve, and all the electronics in that beam splitter will go right here, just like it's shown in that diagram. So within hopefully a month or so, we'll be actually testing these. These do not have a battery pack in them, so they're meant to be a slave instrument where they connect to another instrument that provides power and data logging. And so they're a simplified version, but we're hoping that these will make these much more widely available. That was the goal of the XPRIZE competition, was to make an inexpensive pH instrument that could be widely used by monitoring government agencies and scientists. So we've also, wrap up here, we've also worked on an alkalinity system. Alkalinity is the acid neutralizing capacity of water, and the way it's measured typically is by titration, if you're familiar with that. So we actually do underwater titrations. This has been around. We've had this instrument for a while. We have a paper from 2013 where Reggie Spalding, who works at Sunburst Sensors, deployed it on a coral reef, and we have a couple weeks of data there. First, autonomous alkalinity measurements in the ocean. Again, with this different instrument. This is more like the one-eyed monster, though, in terms of its behavior. It's complicated. It doesn't always do things that are predictable. And so I still have two grad students working on various aspects of atom protein ribosome mesocom, working on improving various aspects of this instrument. And Tunsa Lai, also a postdoc in my lab. We're all still trying to improve this instrument. So this isn't yet commercially available. I wanted to just finish up by saying, so these three papers are these three primary in situ sensor papers for showing that we could put autonomous alkalinity, autonomous pH, and autonomous CO2 sensors underwater. So these were all breakthrough papers, and each one came about, it's coming about 10 years apart, so the next one's going to be in 2026, I think, maybe. But I don't have one that we're working on right now, so I don't know when that's going to happen. So the alkalinity system is one of our main priorities right now. So this has been a really big team effort. Corey Beatty is a longtime technician. He's my lab manager. He's actually returning from the Arctic this week, where we deployed more CO2 and pH sensors on buoys and drifters out there. And so that's a big project that we have, that's a major focus of our lab right now. And then Adam and Reba, like I said, are working on the alkalinity system, and Chunsa also, and Katie is a new grad student in the group. And then, of course, Terry Hammer and some other engineers at WHOI, Woods Hole Oceanographic Institution, were key to making this happen back in the 90s. And then, of course, Jim and Sunburst. And here's some of these folks. And we have had a lot of funding from National Science Foundation, NOAA, and also NOP, which is National Oceanographic Partnership Program, which funded a lot of the commercialization of the instruments. So, thanks. Thank you for time. I guess we have a microphone so we can actually use that, or we can just yell out questions. Steve?
How do you store or transmit the data? How do you store or transmit the data?
Well, most commonly they're stored internally. So I don't have an electronics board in there, but it's just a CPU in there that controls the operation of the instrument and also converts it to digital data that's stored in the memory. In these Arctic systems that are drifters, we're tied into their satellite transmission system, though, because those instruments are not recovered. And so it's expensive to do it, but they have a surface buoy on the ice. We put our instruments through the ice like an ice fishing-sized hole. And then we have a communication device that sends signals up the cable to the satellite transmitter. That's complicated and expensive to do, so most commonly on a buoy, you'll just stick stuff on the cable and then come back and get it later and not transmit the data. And so you run the risk of losing everything. And that's happened numerous times to us, where a fisherman will sweep it away, or it'll break in a storm and be lost, and things like that. Ed?
What's the difference between the pH sensor and the CO2 sensor?
Okay, so I whipped through that, but in that cell that went around, there's that membrane equilibrator.
Yeah.
Well, that's separating the indicator from the ambient pH of the seawater or freshwater. And we have used these a lot in freshwater too, which I haven't talked about. But so that's the CO2 sensor. There's no actual contact of the solution, indicator solution, with the seawater directly. So that pH doesn't do anything to that. Whereas in the pH sensor, we actually pull in seawater and then mix the indicator directly in it. This is a 100-year-old technology that was never automated, basically. I mean, people were making Kolthoff in the 1800s was mixing indicators and measuring pH in samples, but it's just purely automation of that old ancient method that has been the breakthrough. There's no new chemistry or anything.
Are you limited by the amount of indicator solution?
That we use 50 microliters of indicator per measurement. So it's incredibly low volume. This is a big advantage of the technology. We've kept everything really low volume. And so like if you had a cuvette-sized optical cell, for example, you'd be flushing milliliters of water through there, whereas we're just using tiny amounts of liquid. So what do we use in the semi-pHs? How many? What's the volume? What's the total volume of the bag? 500?
Yeah.
500 milliliters will last about a year. So 500 ml bag, like they look like juice bags, will last a whole year of measurements. Measuring every hour, they're not measuring constantly. Yeah. Right.
Mike, to follow up with the pH sensor, do you have challenges at different depths of different ocean compositions affecting your indicator or the performance of the indicator?
Well, it was interesting because during the XPRIZE competition, when we went down to 3,000 meters, there's actually a pressure dependence on the equilibrium constant. This gets into more chemistry than this audience wants, but at those deep pressures, there are thousands of bars, the equilibrium constants all change, and you have to account for that. Jim was out there, and I was sitting here thinking, oh, we never accounted for the pressure in the equilibrium constant. Other people know what it is. So we sent the corrected equation and it made the data. We didn't know what their data looked like, but it changed our data significantly, and then we ended up winning that. So,
Thanks.
so yeah. Mark?
Mike, that's an incredible illustration of a diversity of skills. I'm interested in what part of this whole process was the biggest challenge for you? Probably not the sensor. What I'm interested in, building the whole package that you did, the team, the instruments. What was the biggest hurdle for you?
It was challenging from an academic perspective. Maybe this doesn't directly answer your question, but because that took a long time to get that first 1993 paper out, I didn't have anything to talk about until it actually worked. And then after it worked, I didn't have anything to talk about because it already worked. And so there's a bit of that. I mean, you could publish papers on subtle differences, but I was never wanting to do that. And so I had a sit down with one of the scientists at the Oceanographic Institution, and he said, you're just not publishing enough papers to get on their scientific staff at that time. And that was one of the problems in doing this. So I was way out on a limb. There were 2 years of my hard laboratory work as a postdoc with no breakthroughs, and then and if it didn't ever work, I often comment that I would not. I don't know where I'd be. I might be not in academia if it hadn't worked. So there was a lot at risk there to carry that out. But,
Well, I think that answers it. I mean,
Okay.
the idea of being able to put together the company, the associations, all that, you've emphasized that that's a hard thing for an academic to do. I'm sure it's hard for anybody to do, but.
well, and I never wanted it to take over my life, so I was lucky to get Jim, and we have 9 people there, and it's a self-sustaining operation, and I don't have to worry about that so much. So it's been critical for me to keep my scientific work going. So without that, it wouldn't be what I really planned. First.
So you said you could take a reading every minute or so with the pH sensors?
Yeah, we only run them about every hour, because the resolution, the variability is small on an hourly basis.
Yeah, I guess that was my question. What changes, what time scale do changes take place on that are interesting to you?
Yeah, so you want to resolve diel 24-hour cycles, because a lot of the variability in the short term is related to photosynthesis and the sun cycle. However, in the XPRIZE competition, they lowered the instruments down through the water column, and we had to take measurements. We were taking them as fast as we could, which was, what was it, Ruben? 1 minute intervals. That's as fast as we can go to pump. But that was because it was dropping through the water column, and the changes are steep. In the deep ocean, it gets lower and lower pH because of the accumulation of organic matter and respiration of that organic matter. So it starts out at fairly high pH and goes like that. So if you're not sampling frequently, you miss it. And so that we don't operate it that way very often. The battery power would be consumed fast, but when you're on a ship like that, it doesn't really replace it.
And the alkalinity sensor, you can operate on about that same time resolution?
That takes more power, more pumping, more indicator, more acid. It uses acid as a titrant. And so it only can be deployed on a 1 or maybe 2 hour basis for about a month or 2. So we're trying to improve that. I have a grant now to go to Bermuda and deploy those on a coral reef with another collaborator, and so that's the next step in that project.
So, gathering all this data is absolutely fabulous, and it gives you so much insight. What I'd be curious to know is what kind of analytics do you want to apply to this, and what insights do you expect to be able to glean from having all this information?
Well, that's a good question. So I wasn't able to really go into that, but the data allow us to model the variability. So we can, one of the things I do in my research is when we get these big data sets back, we scratch our heads and try to figure out why it goes up and it goes down and so on, or it goes up and up and up. And so we use models, and we use very simple models here, but by using those models and getting them to predict that variability, gives you confidence that you're really understanding the processes that are controlling that variability. So for example, on a daily basis, you might be modeling photosynthesis, and that's causing the CO2 to go down because plants take up CO2 during the day. And then so you want to model that process, and there's lots of other processes, mixing, warming can affect the CO2 levels. And so if you combine all those into a model and predict the variability, then you could maybe go forward with that model and predict the future. And so given those parameters in the model, like chlorophyll concentration, that's plant matter, temperature, and things like that, wind causes gas exchange, with all those parameters, maybe then you can use that to predict the future CO2 cycle. Our models are very simple. We're giving these data to modelers that have much more sophisticated global models that try to predict the CO2 cycle. So that's really all it's aimed towards, is development of models and then future understanding of what's going to happen to the carbon cycle as global warming occurs.
Mike, it looked like the changes that you see on a daily basis are really small. So how accurate does your instrument need to be?
Yeah, so if you know pH, then the resolution's about 0.002 pH units, and the variability day to day though is bigger. Maybe depends on where you're at. In the coast, it's much more productive, and in the ocean gyres, it's very unproductive, and so the daily cycle is very small. But I would say 0.01 is not unusual then for a 0.01 pH change. CO2 might be, when we went to the equatorial Pacific, the CO2 was changing by 3 or 4 parts per million per day on a 350 part per million background or so. So a very small range of variability, but important to try to understand what's driving it. So 1% or less changes are not unusual that you're trying to resolve.
What's the market like for the instruments? Is it other scientists? Is it government agencies? Who's going to be using besides your own lab?
Yeah, so the question is, what's the market for the instruments? It's mostly a research market, and it's an international research market. So we've sold instruments all over the planet, but mostly to academicians, researchers at universities, but also a lot of government labs buy them as well. We have sold a couple of instruments to aquaculture or oyster farms on the west coast of the United States in Oregon. They're getting more interested in it. They don't have budgets to, the SAMI, the new SAMIs, the black one that I showed, are the cost of a Honda Civic, basically. That's what I was equated to, about $18,000. That's without, that's just standard model. But so the aquaculture industry does not have to pay those kind of prices. But they're realizing that they need to monitor it. They've shown, that oyster larvae are affected by high CO2 levels, and some of the high CO2 levels are natural, but they're being increased even more by this anthropogenic CO2. So they're actually interested in monitoring that much more accurately. We've sold not a huge number of them, maybe a couple hundred of each, over 200 of each, the CO2 and pH instruments. So they're globally dispersed, and I have no idea anymore how many are in the ocean at any one time. Sometime I try to Google and find out who's publishing papers, but when it comes to National Science Foundation wants you to have broader impacts. Because of that, I have good broader impacts. And my reviewers will always say Mike has good broader impacts because he commercializes his instruments and lets us all, and then we can use them. So it's actually, and I didn't realize how much of an advantage that would actually be in terms of broader impacts.
And real quick, did you have to plug the output of these into something like NEON, the long-term ecological observatories that the NSF was supporting?
We're not in a program like that, and so I'm trying to think of a marine equivalent to that. We're part of the Arctic Observatory Network, and so that requires we submit our data within a 1-year time frame to a data repository, and then that becomes available. They have to contact the originator of the data to work out co-authorship and stuff like that, but those data become available within a year's time, and mandated by that program, by this Arctic NSF Arctic program. So they really are forcing scientists now to get the data out off of their computers and into the public domain. And when I had my Arctic renewal proposal, my review said, well, I looked on the ARCIS website and Mike's data was there. So thank God I put it in. You think I'll get to it soon, you know. But, you know.
All right.
Okay, thanks, Steve.